the Pediatric Gene Therapy, Medical Ethics, or the PGTME, as you'll hear today, our third annual lunchtime lecture series. This is our third day this week, and we're really, really grateful to have all of you for this, now our third year of hosting this event. Personally, I'm really humbled and inspired by the backgrounds of the attendees all week, but especially today for our discussion and hopefully thoughtful engagement that's already occurred before, will occur during today, and then after on the discussion of our topic today. For those of you who don't know, the PGTME is a project for within the Division of Medical Ethics that's housed in NYU's Grossman School of Medicine's Department of Population Health. You can learn more about the PGTME on our webpage, and you can also sign up for our quarterly newsletter to stay abreast of what we do. My name is Rafael Escandon, and I've been a member of PGTME since 2019, so about to enter the fourth year. My own background is I've had a career in clinical research and development in the biopharmaceutical industry, and now have a clinical research and development and research ethics consultancy here on what's chilly Bainbridge Island in Washington State. And while it's not quite lunchtime here yet, we do make time for coffee, so alert and ready to moderate, I think, what's going to be a great session. So today's topic is gene therapy in sickle cell disease. And speaking for myself, I couldn't be more stoked or excited to hear from today's panel. I think we've got just such a diverse and expert panel. And as we were discussing a little bit earlier, a year from now, if various regulatory approvals go through, we may be looking at the first gene therapies that get approved in sickle cell disease in the United States, which is really monumental. And I think, as our clinicians know, some of these therapies may be truly transformative in nature. So we'll have to wait and see there, but really excited to discuss the landscape today, and then look forward to what we may face in the future. I'm going to now ask each panelist to briefly introduce themselves, and then following those intros, we'll have a moderated discussion, and I'm going to try to leave minimum 10, but 15 minutes at the end for Q&A. We encourage you, as I know Kara's pushed out already, to submit questions in the Q&A feature here on Zoom, and we'll do our best to get to them before the session concludes. But if we don't get to your question, we'll also do a roundup and make a communication afterwards to address as many as we can. Okay, so I'll start. How about we start alphabetically? So it looks like Sheila, why don't you go first and introduce yourself in a minute or so, and then I'll facilitate that. Lori will go next, and then Lewis, and then Akshay.
Hi everyone. So my name is Sheila Cintron, and I have a background for about 23 years in the healthcare industry as a practice administrator. So I work a lot in the backgrounds with many clinics, but best of all, I'm a mom to two beautiful children who I can say now had sickle cell. So one of my daughters, Helen Obando, has gone through the gene therapy, and my oldest, Helen Obando, went through bone marrow transplant. So vast differences in both. So I want to speak a little bit about that in our experience. And I can't forget my son. He won't be happy if I don't mention him. He was my daughter's donor. So again, been a huge advocate for many years with sickle cell, and very happy to be here and share my knowledge and experience with sickle cell.
Thank you. Wonderful. Lori.
Good afternoon. My name is Lori Crosby. I am a psychologist and professor of pediatrics at Cincinnati Children's, and I'm also a member of the Cure SCI work group on patient readiness and resilience for gene therapy for sickle cell disease. So very excited to share what I am learning and have learned about this process, both from the psychological perspective as well as the stakeholder perspective.
Thank you. That's great. Louis, you're next.
Hi, I'm Louis Su. I'm here representing Sickle Cell Disease Association of America. I'm a pediatric hematologist currently in clinic at University of Illinois, Chicago. And I apologize if we have much background noise. I am not a gene therapy treater, but I refer patients to gene therapy and have been thinking about this for a while. Also from the perspective of the community-based advocacy organization, Sickle Cell Disease Association of America.
Great, thank you. And Akshay next.
Thank you so much. I'm truly honored to be here with all of you. So I am a pediatric hematologist and a bone marrow transplant specialist. And as we move forward, I'd like to call myself a gene therapist also, because that's what I try to do. I'm a clinician scientist at St. Jude Children's Research Hospital in Memphis, Tennessee. And my research focuses on developing curative therapies for children with sickle cell disease, including both transplant as well as gene therapy. In addition to that, I also consider myself a sickle cell advocate. Some of the work that we have also done is surrounding the impression and what people think, especially patients and adolescents think about gene therapy and transplantation, and how do we approach these things? How do we talk about them as we move forward? Because as I'm sure all of you will agree, these are extremely complicated and novel therapies and not very well understood. So how do we make these accessible in the sense that people truly understand what they are getting into? And so that's part of our work, and hopefully we'll talk about that today.
Yes, definitely. Thank you very much, Akshay. Much appreciated, and to all of you for your intros and again for participating. I thought just because there's a diverse background for participants in the webinar today, I wanted to just briefly touch on the biology and the history of sickle cell disease, and I did do a little bit, went back to believe it or not, to the Bible of biochemistry, Streyer's biochemistry textbook. It's been a long time since I picked back up, but just did a little research on hemoglobin and its history in terms of science, biochemistry, science, and medicine. And you know, it turns out that sickle cell disease was actually hypothesized, the cause of it was hypothesized by Linus Pauling about 5 years before Watson and Crick discovered or elucidated the genetic code that was kept in DNA. And I think I want to focus on, just ask this question of the clinicians. Because gene therapy is actually doing a couple of different things, whether it's a CRISPR program or whether it's a gene replacement program, can you tell us a little bit about the difference between hemoglobin A, hemoglobin S, hemoglobin F? I think that's just a basic biological foundation that I think the audience needs to appreciate, because I think the CRISPR and the gene replacement programs target those elements a little differently. So I don't know if Louis or Akshay wants to grab that, some of the basics there.
That's okay. All right. So in each person's red cell is the hemoglobin, and the hemoglobin would come from two genes, one from the dad and one from the mom. And if a person has two genes for sickle, then all of the hemoglobin inside the red blood cell would be sickle hemoglobin, which has the unfortunate property that when it loses its oxygen, it can stack up into large, rigid blocks. And that distorts the red blood cell, makes it fragile, damages it, shortens the lifespan, blocks circulation, causes all kinds of problems. But if inside the red cell you have a mixture of the sickle hemoglobin plus some other hemoglobin, then it interferes with the ability to make those blocks, those polymers. And so you may not have any symptoms, or you may have much fewer. And depending on which other hemoglobin there is a spectrum of different levels of improvement. So if you have normal hemoglobin, hemoglobin A, and sickle, that would be sickle trait inside the red blood cell, that can polymerize, cause the problems under certain very extreme conditions, extreme like mountain climbing while dehydrated, or very low oxygen breathing conditions, or extreme exercise or dehydration. If somebody has fetal hemoglobin and sickle, the fetal actually interferes with the polymerization even more than the hemoglobin A does. So fetal hemoglobin, which is what everybody makes when they're in mother's womb, usually gets turned off as you grow older. But if you can reactivate it, turn it back on again so that there's lots of fetal hemoglobin, or if you can introduce more hemoglobin, fetal hemoglobin production by gene therapy, then that can interfere with the polymerization and makes that red cell not sickle so much. And if you have a mixture of fetal and A and S, then that even polymerizes less and would be even more effective as making the red cell more normal. So that's a short description. How is that, Rafael?
Yeah, really helpful. And so the therapeutic approaches of the gene therapy tend to optimize the expression of fetal hemoglobin. That's what I'm understanding, right?
Yeah, and I can speak to that a little bit. So the therapeutic approaches, as far as genetic approaches are concerned, they primarily focus currently on two different things. As Lewis said, when we have adult hemoglobin, HbA, in the cell, in addition to HbS, sickle hemoglobin, things are better. So all the therapeutic approaches that we are using are basically trying to reduce the sickle hemoglobin and produce some other type of hemoglobin. Now, that can be fetal hemoglobin, HbF, or that can be a substitute of an adult-like hemoglobin. So, for example, some of the gene transfer approaches or gene addition approaches, such as the one being employed by Bluebird Bio and the one that was being tested at University of Cincinnati by Dr. Poonam Malik, those two approaches were taking a slightly modified version of the adult hemoglobin and putting that into the cell. So now the cell, in addition to making sickle hemoglobin, would also make some adult hemoglobin. And then some other approaches, whether that the one that is using a short hairpin RNA, which suppresses a transcription factor, which leads to turning off of fetal hemoglobin. So if you turn off the off switch, it turns fetal hemoglobin on. That approach or other approaches which use gene editing to modify the gene somehow, they upregulate fetal hemoglobin. Ultimately, the bottom line is, let's try to reduce the production of sickle hemoglobin in the cell and make some other hemoglobin higher, whether that is fetal or adult-like, or now there are experimental approaches which are also going to produce another novel hemoglobin called hemoglobin Macassar, which is a normal hemoglobin variant that is seen in individuals in Southeast Asia. And so that hemoglobin Macassar approach is currently in preclinical testing, but it is very likely that in the next couple of years, you'll hear about that in clinical trials as well.
Fantastic. So the upshot here is, if you can change the ratio of the types of hemoglobin that are produced and increase other types other than HbS, that's a reasonable therapeutic approach from a gene therapy perspective, and that can make a difference clinically. Superb. This is great. So hopefully that's enough background for folks who are relatively new or less familiar to this, and it really portends, I think, my next question of the panel, which is, tell me about, and I'm going to direct this to Sheila and to Lori first, I think. Tell me about how well you think, because again, the biology is fairly complicated. The therapeutic approach for gene therapies and gene editing is fairly complicated. How would you how would you sort of describe the level of understanding, obviously for parents first, who have young children who parents are making decisions for, but then as children become adolescents and even into become young adults, what's the level of understanding? What's the level of what are some of the factors, some of the big factors that you encounter in discussing the condition and to treat these very novel and sort of cutting edge treatments with kids and adolescents?
I can speak for myself, having gone through this personally with both of my children. So when this was approached to them, I actually sat them down. So this was not a choice that I made for them. I actually let them make this decision. They went through counseling, they spoke to many doctors, all the risk factors to it was explained. But in my case, I think Helen only heard I may not have sickle cell. So I don't think her understanding was completely there. She understood the risk factors, but didn't understand how it was going to work. I think in her mind, it was, okay, I'm not going to have this pain. I'm not going to have this discomfort. This is possible. So in my experience, again, the way that it was explained, I understood it being an adult and being in the healthcare industry. But speaking to her hematologist, who only does pediatric hematology for sickle cell, he explained it in much, I don't know, cartoonish way for her that she understood it. Just as he would explain what happens, he will always mention how traffic goes through the blood and just explaining it in that way. But I don't necessarily think that they understood what the study is. She just understood that she may not have to suffer anymore.
How old was she, just out of curiosity?
She was 16. She was 16 when we started talking about it, and 17 when she got the transplant for gene therapy.
And then one other quick question, and Akshay and I have talked about this separately. The actual informed consent form and the pediatric assent that she had to, that were part of the process for research, was that 30 pages long? Was that 5 pages long? Was that, how did that, what's your recollection of that?
It was a mortgage signature. It is very, 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 very big. As you know, being a sickle cell advocate, one of the things I did was like, no, I need to take this home. I need to really digest what I'm reading. And that's what I did. It took me, honestly, about a month to really take a look at this, but it was tons and tons of paperwork. And some of them were a little repetitive, but just with different wording. So if that can be shortened and understood, that would be great.
Yes, Lori, this is a great segue for you. And as part of the care team, tell us about your perspective on adaptation of these things in research and families and just that whole dynamic from your side.
Yeah, so Sheila brings up a really important point. There's the education about the actual process and what's going to happen. And then there's expectations. And I think, you know, when we're educating folks, we have to make sure that we do both and we pay attention to both. And so I think there are people who are doing very innovative things with trying to break down the education into cartoons, as Sheila mentioned, into other things that people can really digest the concepts of what's happening. But I think the part that we recommend at least a month, like Sheila took, and having someone to talk to, an advocate or someone else who can help explain things in lay language, is really the expectations and really walking people through the process so they sort of know what to expect. What we've heard from some of stakeholders is that they just weren't prepared for better than the best effects. So they were prepared for a cure, but they weren't prepared for anything else. And that could be because of what Sheila sort of mentioned. You focus on the one thing, and that's sort of the thing you're holding on to. But we want to make sure that individuals really get a full understanding of the process and all of the outcomes or the range of outcomes that could exist, so they're prepared and ready. And so I think our focus on readiness and resilience in the work group that I work on, that's what we're working on, trying to give those tools to clinical teams so that they can prepare and work with families and adults so that they can make a good decision.
Yeah, no, so important. And you've actually hit a couple of threads that I want to pick up on. Maybe kick these to Lewis and Akshay. There is a lot, and a couple of questions have come in. There's a lot that is not known, right? So here's what's different about gene therapies, and I talk about this a lot. We draft off of clinical trials from drug development, but these are very different treatment modalities, right? You get them once, you can't turn them off. We don't know how long they last. We also don't know down the line if there could be sequelae that haven't emerged yet, right? So there are lots of mysteries, probably both good and bad, right? And I think we have to obviously stay focused on the positive and wait for data to emerge. But this is a very different, if you're volunteering for a clinical trial, this is a very different scenario than if you were volunteering for a small molecule trial or even a biologic trial that has a very different half-life. So I wanted to talk about, just maybe from Akshay and Lewis's perspective, how do you think about and how do you present the unknowns, right? And how do you modulate that based on age, based on other factors in this population and others? Because there's a lot we know, there's a lot to hope for, but there's a lot we don't know.
So I can get started on that. And I think Sheila and Laurie hit on some of the critical points that I struggle with. And I should mention that this is not just a problem of the clinical trial participants, but this will also be a problem with the early adopters. When the therapies become approved, because when we think that something is approved, we automatically think that that must be safe, that, we know enough about it. We know everything about it that we need to know. But that's probably not going to be the case for many years, maybe decades. And so part of the reason why those consent forms are so packed is because clinicians like me, for whatever reason, good or bad, good reasons can be we want to provide more and more education. Bad reasons for making consent forms so onerous is that people don't want to be litigated eventually, which should not be the goal of consent form, but that unfortunately has become the goal. Anyway, what I'm trying to convey is that the reason these consent forms are written the way they are and that information is conveyed in that way is because we don't know a lot about these therapies. Transplantation has been around for about 30 years. So we at least know a little bit more about that. Gene therapy is, on the other hand, very, very new. I mean, the patients that have received gene therapy, the longest duration of time, have been less than 10 years. And so like Rafael, you mentioned, there can be both good and bad things. And I think it's this level of uncertainty, how to convey that is the most challenging, because like Sheila, you were saying, when you're presented with an option that, hey, you can do this therapy, it'll last whatever, 3, 4, 6 months, and after that, you won't have sickle cell disease anymore. In all of our minds, there is that therapeutic bias. I want to be done with this sickle cell disease, and then nothing else matters. And Laurie, I'm very scared to use that 4-letter word, cure, because I don't know if we are actually providing that. As you all know, many of the complications that patients have with sickle cell disease before they undergo therapy, they may persist afterwards. If they've had a stroke, that's not going to get cured. You're going to have that stroke for good. If you've had bone necrosis, osteonecrosis, as we call it, that might actually worsen after therapy. And so one of the perceptions that many patients might have is that, if I do this, I am done, I'm good for forever, and I'll not have any problems at all. That may not be true for them, and they may need to continue having regular follow-ups, treatments with physicians or treatments in hospitals. I mean, that may continue. And so I think that's and that's unknown, right? Like there might be a patient who had a lot of complications early on, but they underwent either transplantation or gene therapy and then nothing after that. And then there might be another patient who develops complications of either transplant or gene therapy, or continues to have sickle cell related problems going forward. As of today, I don't know of any tool or any scale that can measure that and can predict that. And so I think there is a lot of that unknown. And unfortunately, none of us are good at predicting unknowns, and none of us are good at explaining unknowns. And the only way we can do that is to have a very frank and honest conversation with our trial participants or our future patients and tell them, look, this is what we know, this is what we don't know. And hopefully they hear both sides of it, not just the part that you're going to be cured of sickle cell disease. Again, when I say cured, putting that in inverted commas, hopefully they hear both sides of that, and then they're able to make a judicious, informed decision about what they're going to do. And the only thing that I try my best is that I don't let them make that decision after a 1 hour conversation. I think, Sheila, what you did was absolutely correct. You take that form home and you think about it and you discuss with your family and friends and go back to your primary care physician, you go back to your hematologist. What I try to do, and I force them, is I meet with them the first time and then I tell them, okay, I'm going to meet with you in 3 to 4 weeks. I'm not going to do this again in between, because you need to have that much time to marinate with this, to think about this. And I tell them, whenever you have any questions, you write them down. I'm going to meet with you in 3 to 4 weeks. We'll talk about this again. And even when we talk about it again in 3 to 4 weeks, we are not making a decision then. I want you to think about it again and again. And we talk about it multiple times so that ultimately you know exactly what you're getting into. Because if things go well, then obviously everybody is happy. But if things go south and things don't go as well, we all need to have that satisfaction that we knew what we were getting into, that we fully thought about that and we were satisfied with the decision that we made, because the regrets, that's what you're going to live with for the rest of your life. As Rafael said, this is not a on-off switch. It's a one and done deal, and it's done for life.
There are, exactly. And Louis, I want to hear from if you have anything to add, but this has been a bit of my struggle as I've tried to put some of the ethical categorization around this, is that, again, in the context of previous drug development, at that paradigm, there are some elements in gene therapy that we really just don't have words for yet. So Louis, please, anything to add?
Yeah, so I think I certainly agree with everything that has been said, and just wanted to throw in a few additional perspectives. One is that for some patients and families, the things that they hear is also colored by how much trust they have in the particular person that's talking to them, so that they met the transplanter gene therapist for the first time, and then they've been meeting with their primary care doc or their hematologist for 16 years, they get a different view of the information. Likewise, they might hear the information from the nurse who's been seeing them or the check-in desk person and feel trust with them. So you have multiple different perspectives and sources of information. And some of what I'm looking at is that, a patient will be hearing about gene therapy from me, and then they go to the movie theater or see Marvel comics, and there's gene therapy or gene transfer making somebody into the Green Incredible Hulk. And they say, well, is that what is going to happen to me? So that things get conflated a bit. Likewise, we're hearing worries about, COVID vaccines are going to be changing my genome, and nanobots are going to mess with my DNA. Is that what's happening in this thing, and that's what the consent is not telling me, and that's why they had to write it so long, is to cover up something. So we have conspiracy, suspicion in some patients where different members of the family may have these worries that are unspoken, and it depends on how much trust they have in the person doing the consent. There was one question I'm going to address partially about trying to have video or phone presentations. So the power of those kinds of ways to do consent was showed to me early on in the bone marrow transplant process for sickle cell, where we were trying to show how bad could graft versus host disease be, which incidentally should not be a problem for gene transfer, right? Gene therapy should not cause you graft versus host disease. But when people are putting together a graphic presentation for bone marrow transplant, depending on which kind of pictures you chose for the graft versus host disease, you could choose the most awful, horrible, gory ones and close-ups and things like that, as opposed to pull the camera back and you get a different view. And that scared a lot of people off, just that presentation, who I think were getting a skewed message because of exactly which choice of graphics there would be. So that the communication power in those kinds of things can be enormous. Maybe that we should have some more, but it can certainly skew the message if you choose it wrong.
I do want to add one thing to that, Lewis, and your point is very well taken, the messaging of videos or how you show a photograph. But I would like to add the power of the spoken word or the messaging. So, this is an example which directly relates to sickle cell disease that I encountered. As many of you might remember, CBS did a 60 minutes episode on one of the patients who was treated at the National Institutes of Health with the BridgeBio Gene Transfer. And in that therapy, they use a lentiviral vector. And so during that episode, when they were talking about the lentiviral vector, the narrator very casually said that the lentiviral vector is a modified HIV virus that puts in a new gene into your cell, and then left it at that, and didn't explain any further and moved on. Now, factually, this is absolutely correct. A lentiviral vector is a modified HIV virus, but if you pull back and think about it a second, so you are telling me that you're treating me with HIV, you're putting HIV in my body. And so when that episode happened, a lot of my patients came and told me and asked me, hey, are you doing that HIV therapy to me, or are you doing something else? Because if that's the HIV therapy, I don't want that. And one patient went on to the extent of saying, again, talking about conspiracy theories, he said, first they gave us syphilis, now they're giving us HIV. Right. Talking about the Tuskegee experiments. Again, we know that nobody gave anybody syphilis. It was an observational study. Again, ethically wrong, but not giving. And again, here it's very different, but this is how the spoken word, the narration matters and creates. So I wanted to kind of pick your brain, Lori and Sheila, about this. How did you navigate that?
So I can definitely speak to this. So we were interviewed for the New York Times for, I want to say it was like a year and a half, almost 2 years, with cameras following us everywhere. It's actually up on Hulu, where they have the whole documentary on Helen and how the process went. And one of the biggest things that they touched on is how this happens. And they do mention, the vector with HIV, but one of the things that I pulled back on, and I said, look, we need to explain this a little more, because for some outsider looking at this, they're going to, okay, am I going to have HIV now? So they went back, re-edited the entire process to add as to why that happens, and it's really not the live virus. They explained it all. Even with cartoons, actually, they explained it all. So that's a huge point. Not speaking thoroughly of how things work, especially, bringing another virus that's potentially devastating to anyone and not explaining it, can be a shocker, can raise more concerns. And, especially with COVID, with all these conspiracy theories on the vaccines and what we're doing and it's invented and whatnot, is very, very important. So again, I would recommend, if you watch that documentary, it really breaks it down. And, being a part of that and having lived with children with sickle cell, I again was once again an advocate, like, okay, hold on. I don't want people to misinterpret what gene therapy actually is into something completely different and discourage people from doing any type of gene therapy.
Exactly. And it just takes one, as you say, it takes one little crumb of doubt to just mushroom into a lot of conspiracy. Lori, I want to ask you a very specific question. I want to actually pivot a little bit now, because Lewis and Akshay both mentioned interactions with primary care, and I think this is a very important part of gene therapy and pediatric gene therapy in particular. And I'll just set the stage for, it's a little bit of an elephant in the room, but two things. The 5 now gene therapies that are approved in the United States are all for very, very rare conditions, much less frequent in occurrence than sickle cell disease. And so I think as we look down the barrel now of a potential approval or 2 coming in the next year or so, all going well, we have a population that demographically is pretty specific in terms of having sickle cell disease, and that demographic tends to have insurance coverage or state assisted coverage more proportionally. And so as everyone who's been paying attention has seen these each of these successive gene therapies as they've been approved over the past 5 years, 7 years, they just get more and more and more expensive. And so I'm just thinking about what's your team thinking about in terms of trying to coordinate or trying to have the discussion and educate on a state-by-state basis or on a person-by-person, insurance covered by insurance coverage basis about what's coming down the pike. I know it's a complicated question and a lot of unknowns because we don't know how much it's going to cost and what state Medicaids may cover versus what other payers may cover, but it's a reality, right? We may really be coming into a collision here of a transformative treatment being available, but proportionally, most patients with the condition can't afford it.
So thanks for bringing up that issue. And I just I'll try to tie what we were already talking about to this too, because I think 2 things that I want to pick up on that were said by Lewis, Akshay, and Sheila are trust and transparency, right? And so this population, who primarily in the United States is Black and Latinx or Hispanic Latino, has, a history in the United States of being minoritized or marginalized. And so there is an issue with trust. And there's, people like to say historical trust, but it's current. When you have Serena, Williams trying to get the care she needs after a baby, it's current. It's not just in the past. So people talk about historical, but let's just be clear, it's current as well. And so there are a lot of trust issues. The transparency helps. So everything the other panelists said about being transparent is key to building that trust. And in that awareness of minoritized and marginalized populations, we also have to be aware of the barriers against receiving treatments, such as costs that you mentioned and access. And those are 2 really important barriers that we can't ignore in this conversation. So thank you for bringing them up, but I just want to tie that together. And so I think, one of the things that we're trying to do in the Cure SCI is really talk about the readiness and the resilience. And part of that has to be these conversations, not only with the medical and clinical teams about expectations and the education about the process and the burden, but also about what happens, is it accessible? What will be the cost, and what will be the cost, even if you're in a clinical trial? What are the other costs that may come up, and who's going to pay for those, and thinking about that up front and talking through that up front. And so I think all of that information that we're gathering, we're hoping to put together to be able to then take that to those insurance companies, to those state Medicaid offices, and say, here's what we've gathered, here's already some of the gaps that exist already before we have these expensive treatments ready. What are we going to do to cover the gaps and to prepare for these expensive treatments? So I think it is a conversation we're trying to prepare to have.
I mean,
and some people are already having, not me personally, but others are already having.
But we went through the complicated biology of the condition. We went through the complicated biology and genetic engineering of the potential treatments. And now here we are, and the complicated social interactions relating to trust and transparency and consent. And now here we are, another now fourth or a fifth dimension of difficulty, or at least opacity, and a lot of unknowns around payment, reimbursement, and access. And so I think that's anyone else have anything to add or want to comment there? Lewis, bring it on.
Oh, go ahead.
Go ahead.
So I do want to add, definitely the expense is very real. It is very, very real. Sometimes we don't think about who's taking the time off work. How are you going to maintain a home without a job? This is something you're getting into that it's not just devastating the person who's getting this transplant, but the entire family as a whole. And then there's some other aspects too, which I did. And again, I advocated so much for this. And one of the biggest things, my daughters want to become mothers. I had to figure something out in order for me to pay for their storage, for their ovaries to get removed so they can store them for the future. That's another expense added there. So it's very expensive, and I just can't wait for the day that it can all be incorporated in one, and look at the factors, having this disease and not having it, and the benefits of it, and how important it is, even if it costs $1 million, but this is still going to save you such more tons of money, especially having the insurance understand this is the big thing. And I tell you, it took me about 6 calls calling the insurance until I got it covered. Because not everyone in the insurance knows all the ins and outs. So I just say advocate and be persistent. So big point here, financially, it is a huge burden.
Thank you, absolutely. Lewis?
Yeah, very much agree with what Sheila was saying. One other thing about costs is that there is this self-appointed watchdog organization, ICER, who I imagine this group has probably talked about before, has taken on various topics. Yeah, there we go. Thank you, Kara. Various topics and sickle cell, and has, to me, not done the most transparent or balanced assessment of how things are. So that costs could be just the medical system costs. It could be, medications and hospital bills, but it does not include the things like time taken away from work, lost productivity, lost education, and all those sorts of things that could be improved and be on the plus side for balancing out this large, costly one-time procedure. The other is that we have this screwed up medical insurance system where people rotate their insurance coverage every year, potentially, so that the insurance company that pays for this may not be the one that gets to reap the benefits of their investment, they don't get the return on investment for the rest of the time because somebody else could be covering that patient by then, or they could be paying for the next catastrophic cost. So all that needs to be worked out. It's way beyond the scope of sickle cell organizations or sickle cell advocates to fix the whole US medical system.
Yeah, definitely. I think it is a conundrum. I think just to pick up on two threads, and then I think we'll get into some of the Q&A. One thread, of course, is I think both Lori and Sheila mentioned this, participation in a clinical trial, especially a gene therapy trial, with the long tail that gene therapy trials have. That's a family affair, and it impacts every member of the family, whether it's a child and they're old enough to drive or not, they have to be transported to clinic visits. I think there are whole other webinars we could do about integration of decentralized clinical trials, or we've gotten a lot better at those in the past since the days of COVID. But we still have a long way to go in terms of decreasing patient and family burden, participant and family burden in clinical trials, because the burdens are different. They're the same in some respects, but they can be different for every family. So these are things that I think as sponsors of clinical trials, we want to keep in mind. And I think really it's been only in the past 5 to 7 years that we've really appreciably thought about those things. I think regulators as well need to think differently as far as gene therapies go. Again, long tail, with rare conditions, of course, we've done a lot of work to try and get regulators to modify their thinking about placebo control, about lots of other trial design elements that may just take a new treatment out of feasibility, for example, if you have to do a traditional placebo-controlled trial. So there's a lot of work to do in that regard. And I think, and then on the informed consent side, again, I think we can't kind of emphasize that enough in terms of, I think these potential treatments have enormous potential and transformative potential, but we don't really we can't use the word cure. It's not appropriate to use the word cure, but it's not necessarily a temporary fix either. It's kind of longer than potentially something temporary. So I'm trying to be hedge on absolutes here, but I think there's a language, there's a nomenclature I think that needs to be developed a little bit better about things like this. And then finally, on the payment side, on the access side, again, I have a lot of fear that we're about to have a big collision in our society when if these gene therapies become available, because we may see at scale now, even though sickle cell disease is rare, it's not nearly as rare as some of the others, and so there may be a real collision socially in terms of access and equity. And that, as a society, I'm not sure we're prepared. As Lewis said, certainly the healthcare system as it is in the United States isn't prepared to tackle that. Any comments? Anyone want to take? I know that's heavy, but I think it's a real reality that we need to face here, and I want to hear what folks are thinking.
I'll just say one thing, and I think I'll probably get yelled at for saying it, but I would just say that the price point at which most of these gene therapies are currently being marketed is just unaffordable. Like, this is unsustainable. This is no way that this is, at least I can't imagine that at this point it will be equitable to majority of the people who need it. So I think something definitely needs to be done about it. And I'm sure there are people smarter than I am who are thinking about it. I'm just a transplanter. I only do the process. Somebody else needs to figure out the details of how to do it.
I think the point Lewis brought up is very important in terms of reimbursement, right? In this country, we do cycle from provider, from provider to provider, or from payer to payer, right? I mean, I think the average time with insurance plan is about 2.5 or 3 years for individuals in the United States. If you change employment, you often change your payer. So that does complicate the reimbursement for potentially transformative therapy that could last for 3, 5, 7, 10 years, 20 years maybe. So that these are all complications, but as you say, Akshay, we can get some smart people working on that.
Yeah.
I think the other thing that we ought to consider is that, you know, where is do the old models of pricing, as you point out, do the current models of pricing really are they that thoughtful about again the social impact of this what we're facing here? And I think the answer is 3.5 million a pop for sickle cell. I don't see it. It just doesn't seem doable.
Yeah, that's exactly my point. And just to add to this issue, we also have a concern. Well, I shouldn't say we have a concern, but we don't have a proven durability yet. And so now if you're charging $3.5 million upfront for a therapy that has a long tail, there has to be some sort of a, buy now, pay later kind of a program. What if you don't like the product? Can you return it? You can't return it, right? So can you, if the efficacy fades off, and some patients unfortunately need to go back and need additional therapies related to their underlying disease, would they get their money back? Would there be a refund, or would there be a staggered payment model that, if it doesn't work, your payment is split into 5 installments over 10 years. And if in 10 years you start having symptoms again, that you don't pay the last 2 installments, something like that. I guess, again, this is complicated, and I don't pretend to know all about it, but we have to consider all these things, not just of course, the social impact upfront is important, but and also how are the long-term durability concerns of some of these therapies going to be taken into account.
Yeah, I think there's a question in the Q&A about that, and I'll kick it to you and Lewis. Just, again, I know it's different from product to product, but there's a question about insertional oncogenesis, particularly with lentiviral vector-related treatments, and I guess I don't know, Akshay or Lewis, if the answer is the concern over insertional oncogenesis varies from product to product, right? Or varies from approach to approach. Is that kind of all we can say?
Yeah. So one thing that I would say is that the 2 patients that have been disclosed to have developed a myeloid malignancy after undergoing gene therapy, in both of those patients, the vector was not implicated. So we cannot say that the vector led to leukemia in either of those patients. Multiple investigations have been done, and they've completely ruled out the vector involvement. Having said that, though, we do know that individuals with sickle cell disease, especially those who have more severe sickle cell disease, and the longer they've had sickle cell disease, they might be at a higher risk of developing myeloid malignancies. As some of you might remember, the very first patient to get a transplant for sickle cell disease actually got a transplant because she had AML, she had acute myeloid leukemia, and she got the transplant for leukemia. And then we figured out, hey, if you transplant the same way as you transplant leukemia, you get rid of sickle cell disease as well. So there might be an inherent predisposition, and we know a little bit about it, but we don't understand that very well. And then anytime you take somebody's cells out from their body, mess with them, and then put them back, doesn't matter what you mess with them with, whether you do CRISPR or you do lentiviral integration or you do something else, base editing or some other novel therapy which is yet to be discovered or developed. I do think that the process of taking these cells out, messing with them genetically and putting them back does create some increased stress, which could potentially lead to development of myeloid malignancies. Now, again, that risk is not fully understood yet. There are a lot of people who are trying to figure that out. And so, again, these things will take time to fully understand. But all we can say right now is that, there is a risk, and we don't know how to quantify that risk.
Yeah. And and it's all speculative right now because it's hard to really do an experiment truly to test that. But coming from a totally different perspective, At a recent Sickle Cell Disease Association of America convention, we had a whole hour discussion about why some people are choosing not to go for gene therapy. And it was individuals with sickle cell stating that from their perceptions of how the risks, the unknowns down this path compared to the lousy path of sickle cell disease itself, they would rather choose to continue with what they know. The therapies that have been, like hydroxyurea has been already around for a long time. And so that there was this tendency to figure out your own comfort with risk and unknowns as the way to work out this ethical and big decision challenge. But where your care is could make a big tipping of the balance. So that if somebody was coming from, say, Jamaica, and especially rural parts where it's hard to get to the doctors, hard to get services, etc., compared to somebody who is like well insured, well resourced in the United States, they can get anything they want covered by the insurance, you would have a very different picture of what their sickle cell disease course without gene therapy is. And so some of that range of decisions is speaking again to the heterogeneity of sickle cell disease.
Yes, definitely. This has been just fantastic. I want to try to address the one other question. It was touched on a little bit before, but love Sheila and Lori's perspective on this as well. Is even if these gene therapies and gene edits are truly transformative, we don't know how long they're going to last. And in turn, but the disease, as several of you have pointed out, doesn't go away completely. You still may have sequelae from prior issues, whether it's a stroke or whether it's other even transplant related things. And so Sheila, for you and Lori, in terms of preparing families and patients, do you feel prepared for that that nuance, if you will, the difficulties, for example, in the long term that may show up? And did you feel like there's security and a bit of a safety net there? And if not, how do you explain it? And I know we're short on time, but maybe we can sort of briefly touch on that one.
Oh, that's a good question. So I would say after, they don't talk about the after. I'm going to be real. The after is really not talked about. So right now, Helen, she's had death of her hip before transplant. She did get it done. She got surgery, did well. When we moved from Boston, Massachusetts to Arizona, then she started developing the same issue on her left hip. I wasn't prepared for that. I really was not prepared for that. So that's something they do not talk about. Just because this seems to be working, it doesn't mean that other complications that she already had are going to persist. And today, I learned something new, that after having these therapies, death of the bone is more permanent to her, and this is what's happening to her right now. As it was mentioned, that the two individuals who had leukemia, the way that was pretty much, it was like a bucket of cold water. I received a letter instead of receiving it from a doctor letting me know, hey, look, this came up. I wasn't prepared for that as well. So they don't talk about the after effects. They only talk about the moment and what's going to happen during that process. So I've done a lot of digging afterwards after receiving
an area for improvement, to be sure. It sounds like.
Absolutely.
I'm so sorry that we're out of time. I just want to thank each of you again for your just incredibly thoughtful and useful perspectives. I think we could go on for another hour or two, and I would love to. But I look forward to next year, as I mentioned earlier, that this may be a very different, very different discussion next year. So let's wait and see there. Also, so again, huge thanks to the panelists today. I want to point out that we have 2 more days this week where we're going to have lunchtime lecture series. Tomorrow, there's an Ask an Industry and Regulator member. That should be very interesting. Got some great folks on that panel. And then Friday, we've also got a whole session on asking a family member, where I think we can pick up on a lot of the threads from today about how gene therapy and participation in trials affects the whole family and individual participants. There will be an individual participant on that panel as well. So much appreciated, Kara. You've just and John, you've just done a fantastic job here coordinating this, and much appreciated again to all the panelists. We'll stay in touch and hopefully have a really interesting session a year from now.
Look forward to it. Thank you.